David got it to go mother and I woke up and those are back. And you just let me stay there and everything. I'll use my green pen. No, I wasn't expecting that. You're cute. I feel like when you see the camera, it's like rule over the Fritos.
Okay, let's get started. It's gut check time for some of you. First exam results were just all over the place. We had several scores in the high 90s and even over 100. We had some scores that were really, really low. I was really distressed by the number of people who just left 10-point essays blank. Questions about the basic things like the lysosome, which was a study guide question. Things like the other 10-point essay on active transport and facilitated diffusion and differences in those. Those were study guide questions. So some of you need to change how you prepare. You've got a chance to recruit points on this exam. I've stapled to the front of the exam different sets of instructions depending upon what your score was, involving test corrections, making some analysis of what you did and what you'll have to do differently. The worst thing you can do right now is to get down on yourself and give up. Okay? How you respond to this right now really will be very important. Go through the process of doing the corrections. I posted an answer key, so you can compare your test to those. What I'm really looking for on the multiple choice questions is sitting down and going, why did I miss that question? Okay? What was the concept I didn't understand? Or did I misread questions? But I want to kind of an analysis on your part of why you missed questions, and then comparing your essay answers to those that I published and seeing where you fell short. There's a lot of time left in the course. There are a lot of points in the course, but you've got to make adjustments now, and there isn't a whole lot of time until the next exam. Okay, there's another exam coming up in basically 2 weeks, and that'll cover the chapter that we just started last time before this exam on introduction to metabolism. We're going to continue to work on that today and Friday, and then cellular respiration. So if you want to do something to really start preparing yourself right now, start going through those study guide questions with your notes, with your PowerPoints, and start preparing early for this next exam. If you didn't do well and you want to come in and talk about advice about strategies that you can use, changes you can make, please come in and talk to me, okay? I'm not going to require you to do that, but I would hope that you would choose to do that. Also posted on the website, it's been there since the first day, is a set of kind of instructions about my suggestion about how you should study for this course. If you haven't looked at that, I would recommend that you do that. Okay? So shake it off if you didn't do well, but by the same token, make sure that you make the necessary adjustments so you don't find yourself in this particular position again. If you don't change the way you studied for this exam, you're probably going to get the same kind of performance. Okay? If you did do well and you studied for this exam a particular way, it probably worked for you, and continue to do that. Okay? Yes, sir.
So on the agree and disagree part for C and D, I was just confused. Like,
Well, okay. Read over the answers that I wrote for your on the key that I sent you. Okay? And then if you have questions about that, come and see me and I can go over it. Because I graded all 49 yesterday, and I could have missed something as I'm going through it, but look at the published answer first, basically, and compare yours. So one question, the first question ended up being a bonus question because I didn't realize that the slides that dealt with humoral and cell-mediated immunity didn't make it onto the website, and so I didn't want you to hold you responsible for that, so that ended up being a bonus question.
that answered my question.
Okay. Well, nobody missed any points on the first question, but you could get some bonus points on it. Okay, we do have a quiz Monday. The quiz Monday will be on enzymes, which we will probably talk about on Friday. We're going to be doing a 3-week lab in enzymes coming up. And so this is really good timing in terms of correlating what we're doing in class and what we'll be doing in lab. So pages 125 through 132, quiz Monday. And then next exam, Friday, March 20th. That is the Friday before spring break, not the week of spring break. But we do have a lab quiz on spring break, okay? On that week before, sorry. So I didn't want to load you up with a lecture exam and a lab exam the same week. And I know that a lot of classes are having exams the week before spring break. So you will have a lab quiz, but you will not have a lecture exam. That's the Friday prior to it. Okay? Okay, today's material is probably some of the more complicated and hard to wrap your head around material that we're going to cover, especially when we start talking about exergonic, endergonic, spontaneous, non-spontaneous, free energy, etc. So please keep alert today, okay? I'm going to try to go through at a reasonable pace and make things as logical as I can, but keep with me. If you find yourself dozing, don't be afraid to get up, walk around a little bit something, but we really need to have your attention today, okay? You don't want your attention every day, but this one particularly can be a little bit hard to follow. So, we talk about ATP as being kind of the energy currency of the cell. A form of chemical energy that the cell can spend to do work. And hopefully you understand now that if a cell doesn't do work, it doesn't stay alive. Otherwise, that second law of thermodynamics that says that everything is going to become disorganized makes cell death inevitable unless you're expending energy to counteract that natural tendency. And at the end of the period, we talk a little bit about how ATP hydrolysis, which is the conversion of ATP into ADP and phosphate, really is responsible for driving this cellular work. And one of the big things that we talked about is that hydrolysis releases this phosphate group. And then when that phosphate group phosphorylates a molecule like a membrane protein, it changes its shape. And that is what was responsible for a lot of the cellular work. So motor proteins, for example, transport proteins, and then we'll get into this idea a little bit later on as we talk about cellular metabolism, is that ATP can actually drive metabolic cellular reactions. So, where do we get our ATP? Well, it's produced by a complex biochemical process called cell respiration. And we're going to get into cellular respiration next week in detail. But think of it this way. Remember that I said that organic molecules that had hydrogen on them were really sources of potential energy. When you see a molecule that has hydrogen on it, think of those hydrogens as being potentially sources of energy. In some respects, those molecules are kind of like a checking account. And that checking account can be cashed out into cash by this process of cellular respiration. So a molecule like glucose, C6H12O6, has a lot of hydrogens. That represents a store of energy, a check if you like, that can be cashed out into ATP by this process of cellular respiration. Now, when we talk about metabolism, it's really important to be able to distinguish between these two terms, autotroph and heterotroph. And I can pretty much guarantee you right now, there will be a question on the next exam that asks you to differentiate between these two strategies or those two terms. Okay? So autotroph, heterotroph. Does anybody know what autotroph means?
It makes its own food.
Okay, literally it means self-feeder. This is an organism that makes its own food, and very specifically, the most common kind of food that autotrophs make is what? Anybody know? Glucose, and it's made by the process of photosynthesis. Okay? In photosynthesis, which we will talk about after the second exam, a couple of inorganic molecules are converted together to form carbohydrate, glucose, and oxygen as a byproduct. Okay? So these are the organisms that make their own sugar, but then what do they have to do with that sugar to make ATP for cellular metabolism? They have to burn it in cellular respiration. Okay? They have to then cash that check. So they make their own check. Okay? What does heterotroph mean? They get their food from other sources. Hetero means other. So they feed on others. Are we autotrophs or heterotrophs? We're heterotrophs, right? Plants are autotrophs. Okay? It'd be kind of cool if we could be autotrophs. Okay? Walk out in the mall and soak up some sunlight and make your own glucose. We can't do that. Okay? Where do we get our food energy? Yeah, we eat stuff. We eat plants, we eat animals. And when we eat them, what are we doing? We're consuming their tissues. And what are tissues made of? Cells. And what are cells made of? Molecules. They're made up of carbohydrates, lipids, proteins, nucleic acids. So we ingest those molecules and then burn them in cellular respiration, cash the check, and make ATP. Okay? Do autotrophs require heterotrophs to survive? Could we have a plant-only world? Sure could. Okay? Because they capture the sunlight energy, make their own food, and then use cellular respiration to make ATP. Can heterotrophs survive without autotrophs? Because they can't capture the sunlight energy. They have to get their food from another living organism. So plants are the producers. Animals and other heterotrophs are the consumers. Well, the process of cellular respiration, we'll see later on, is a fairly complicated process. And what it involves is a sequential conversion of glucose into something else, into something else, into something else, into something else. Eventually, during cellular respiration, when that glucose is broken down, typically in the presence of oxygen, but oxygen doesn't necessarily have to be present. What are the byproducts of glucose breakdown? What do we produce?
CO2.
We produce CO2. We produce water. And we produce energy. What are the forms of energy that are produced?
Thermal.
Thermal energy, good. Heat. And your body has to radiate away that heat or it's going to cook. But if things are working properly, we also make ATP. Okay? So the energy of chemical bonds in glucose is ultimately converted into heat energy and the chemical bond energy of ATP. This is what the cell can spend to do work. Can the cell spend glucose directly to do work? No. Just like you can take a check into some places and they might not accept it for goods. But if you could go to the bank and cash that check, everybody takes cash. So the cellular respiration process is the check cashing process of the energy system. And it happens in several sequential steps, each of which is regulated and controlled by a particular enzyme. Okay, so let's talk a little bit about metabolism. Metabolism refers to the sum totality of all the chemical reactions that occur in the cell. You take all the chemical reactions and lump them together, that's metabolism. So cellular respiration is one example of a metabolic pathway. Photosynthesis is another example of a metabolic pathway. And there are a whole bunch of other ones as well. And again, it's important to understand that cellular respiration and other metabolic pathways are arranged in these sequences of step-by-step conversions. For cellular respiration, that's really important. Because during this step-by-step conversion, small amounts of energy are released that the cell can harness. If cell respiration just went boom, glucose to CO2 and water, it'd really be hard for the cell to harvest the energy that was present in glucose. Let me give you an analogy. When you get out and want to start your car and drive, one of the ways you could make that car move would be to open up the gas tank and throw a match in it. Car would move, right? A little bit, and then it would have exploded. The way cars work instead in the internal combustion engine is that you've got some kind of a device, used to be a carburetor, now it's a fuel injector, that squirts little bits of gas into a cylinder, and there's a tiny little explosion that drives a piston and then drives a crankshaft and causes you to move. With those little controlled bursts of released energy. That's kind of the strategy that the cell uses in metabolic reactions. Convert one thing into another, another thing into another, another thing into another. And along the way, hopefully, little bits of energy are harvested, typically in the form of ATP. Well, there are two basic types of metabolic pathways. The first are catabolic pathways. And again, I can pretty much guarantee there will be a question in the next exam asking you to differentiate the characteristics of catabolic pathways. What are they? Well, catabolic pathways take things that are complex, like glucose, and break them down into smaller units, like carbon dioxide and water. So they degrade complex molecules into simpler ones. These reactions are energy releasing. So we start out with something that has higher energy, and we end up with things that have lower energy. So that conversion is considered to be downhill. And what happens to that energy that's released? Hopefully the cell is able to harvest it or trap it in the form of molecules like ATP. Typically, these particular reactions are oxidation reactions. Okay, time to talk about oxidation and reduction for a second. What is oxidation? What happens to a molecule when it's oxidized? You know what that stands for? Lose electrons, oxidized. If you lose electrons, you become oxidized. What does that stand for?
Gain electrons.
Gain electrons become reduced. So molecules that lose electrons become oxidized. Molecules that gain electrons become reduced. These reactions happen in pairs. So if something becomes oxidized, something becomes reduced. Now, in biological systems, oxidation and reduction reactions typically involve hydrogen atoms as opposed to free electrons. Sometimes free electrons are involved, but oftentimes you're gaining or losing hydrogens. Okay? Well, what is a hydrogen? It's an electron and a proton, right? So if you lose a hydrogen, are you losing an electron? Yep. If you gain a hydrogen, are you gaining an electron? Yeah. So what's happening to glucose? During cellular respiration, glucose is becoming oxidized. We're splitting hydrogens off of it. And what happens as a result? Carbon dioxide is formed. So glucose is oxidized into carbon dioxide. Is there an accompanying reduction occurring? Yes. Where do those hydrogens go? Oxygen becomes reduced into water molecules. Okay, so these reactions are paired together. In photosynthesis, carbon dioxide picks up hydrogens. So it becomes reduced into sugar. What happens to water? It loses hydrogens and becomes oxygen. One of the things that I hope you can come to appreciate about metabolism and cell respiration, particularly, is that it's really all about moving hydrogens and electrons around from one compound to another. Oxidizing one molecule, reducing another. And in the process, energy is generated that lets the cell do work. So typically, the substrates in these catabolic reactions are oxidized. Hydrogens are ripped off of them. These reactions are spontaneous. We'll get there in a second. We'll talk about what it means to be spontaneous. Second type of reaction. These are anabolic reactions. Anabolic reactions tend to take simpler units and stick them together to make something more complex. Okay? So simple molecules like carbon dioxide and water can be stuck together to make a more complex molecule, glucose. These are energy requiring reactions. These were energy releasing. These require an input of energy. What's the input of energy that we need to drive this anabolic reaction of photosynthesis? Where does the energy come from?
The sun.
Sun. Typically, in this particular reaction, we've got a substrate or a starting molecule that's being reduced. So in photosynthesis, CO2 becomes reduced into glucose. And these reactions are not downhill, these are actually uphill. So CO2 and water energetically are down here. CO2 and water don't have much calories, right? You can inhale all the CO2 you want to, drink all the water you want to, and it doesn't have calorie content to it. But glucose is up here. Okay? It's got calories. So this is an uphill reaction. It requires an input of energy. And that's not spontaneous. Okay? The natural tendency of matter is to go here. It is not the natural tendency of matter to go from here up to here again. This is a kind of an abbreviated roadmap of some of the key metabolic reactions in a cell. And each one of these little dots represents a compound that's then being converted into something else, being converted into something else, something else, something else. There's a chart on the door of the cell biology lab that's falling apart that shows you in a little bit more detail some of those metabolic pathways and shows you the names of some of these compounds and some of their structures. But it looks pretty complicated, doesn't it? It's almost like a street map. And these pathways oftentimes are interconnected, just like streets are interconnected. And sometimes the cell might want to do this reaction. But other times the cell might want to do this reaction. Or this reaction. So one of the things we're going to talk about is, how is all this controlled? How does the cell regulate all this stuff? I'm not quite ready there yet. This is a very central pathway in cellular metabolism. Anybody recognize what this might represent? This is a set of reactions called glycolysis, and this is Krebs cycle. Okay? Those are two very key parts of cellular respiration. Some of the reactions take complex molecules like glucose and break them down into simpler molecules like carbon dioxide and water. But some of the reactions take simpler things and stick them together to make more complicated molecules. So you might take a glycerol and some fatty acids and join them together to make a fat. Okay? So there's this kind of counterbalancing anabolic metabolism, catabolic metabolism. Overall, the totality of that is just called metabolism. Okay, so we said that catabolic were spontaneous, anabolic were not. So what makes a reaction spontaneous? They happen on their own without an input of energy. Basically, they're the result of the second law of thermodynamics, which says that all matter in the universe tends to become more disorganized or tends to achieve a higher state of entropy. Remember entropy from last week? Entropy is disorganization. So here are some examples of spontaneous reactions. A diver diving into the water. Concentrated molecules going into more spread out or dispersed molecules. Complex molecules being broken down into their simpler units. All of these are spontaneous reactions. Why? Because they're all going from states of high free energy to low free energy. Or if you prefer, they're going downhill. That is the natural tendency of matter according to the second law of thermodynamics. Everything goes downhill. Okay, here's where it gets a little bit hairy, so hang with me for a second. Okay? So we're talking about chemical reactions. Let's say that we're talking about this generalized chemical reactions where A and B are two different molecules and they're interacting together to form two different products, C and D. So A and B interacting and then producing a new set of products, C and D. We call these reactants or substrates. We call these products. What does the double arrow indicate here? It's potentially reversible reaction, okay? So we can go this way, but we can potentially go this way as well. Okay? Spontaneous reactions proceed with a decrease in free energy. They go downhill. Reactions that aren't spontaneous need an input of energy. They go uphill. So if the reaction written in this direction is spontaneous, what's the reaction written in this direction? It's not spontaneous, right? If the reaction written in this direction goes downhill, then the reaction written in the opposite direction is uphill. This one releases energy, this one requires energy. Well, to dig into this idea of spontaneity and energy a little bit, we need to define what free energy is. And basically, free energy is the energy available to do work. Not all energy in the cell is available to do work. Low temperature heat energy, for example, is not really available to do any significant work. Chemical bond energy, for example, though, is able to do work. It's available to do work. So there's different kinds of energy. Some can do work, some can't in the cell. The maximum amount of free energy that can be harvested from a reaction is the free energy change of the reaction. It's the difference between the free energy of this side and this side. And we call that delta G. If you want a mathematical definition of this delta G, it is equal to delta H minus T delta S. Heck is that? Okay. This represents your free energy, the energy available to do work, to keep the cells alive. This represents total energy. This represents energy unavailable to do work. Basically, this is the entropy component. I'm not particularly interested in you completely wrapping your head around this right now, but I wanted those of you who will see this in chemistry later on and in other courses to have at least seen this one time. But right now, for our purposes, just think of free energy as the energy the cell can use to do work. And spontaneous reactions proceed with a decrease in free energy. Not spontaneous reactions proceed with an increase in free energy. Spontaneous downhill, not spontaneous uphill. And think about a rock sitting at the top of a hill. It spontaneously can roll down that hill and in the process release energy. But if you want to get that rock back up to the top of the hill, you've got to push it back up there. And that's going to require an input of energy. Well, how do we determine the free energy change of a reaction? Okay, so we've got this reaction up here. A plus B going to C plus D. How do we know what the delta G of that reaction is? Here's where it gets a little bit confusing. When we want to determine the free energy change for a reaction, we take the free energy of the products and subtract from that the free energy of the reactants. So the delta G for this particular reaction would be the free energy of this minus the free energy of this. That's a little counterintuitive, and that's where you mess up sometimes. So, what happens if the free energy of the reactants is greater than the free energy of the products? Free energy of reactants, products. Okay, is that be spontaneous? Yeah, downhill. What will be the delta G of that reaction? Negative, because we take this value and subtract this value from it. Does that make sense? So if I've got 10 units of energy down up here, and 5 units of energy down here, what's the delta G? 5 minus 10. That's a minus 5 delta G. If the delta G is positive, then the free energy of the reactants is less than the free energy of the products. It's not spontaneous. That's uphill. What if delta G is 0? Then that means there is no difference in the free energy of the products and reactants. If that happens, the reaction is at equilibrium. And a really important thing to understand about that is, if delta G's are 0, no work can be done. Because delta G represents the energy available for the cell to do work. So if we let a reaction go until its delta G is 0, can't do any work. Okay. So higher free energy, lower free energy. Less stable, greater work capacity. More stable, less work capacity. Delta G of this reaction would be 0 because we take this and subtract this from it. Well, another way we can characterize metabolic reactions is to refer to them as being exergonic or endergonic. And again, as you probably could guess, this is something you will have to know for the next exam. There will be a question we'll ask you to determine the differences between exergonic and endergonic. This has to do with delta G values for a reaction. What's an exergonic reaction? It's an energy-releasing reaction. If we look at this particular graph right here, this axis represents the free energy of molecules. So as you move up on this axis, free energy gets higher. This represents the progress of the reaction. So over here, everything is reactants. By the time you get over here, they've been converted into products. Okay? In this particular type of reaction, the exergonic reaction, the free energy of the reactants is greater than the free energy of the products. Here's our free energy of reactants right here on the axis. Here's the free energy of our products. Okay? So this is going to be an energy-releasing reaction. This is going to be a downhill reaction.
So would that mean that catabolic reactions are also exergonic reactions?
Yep. Yep. We're getting there, but that's a good conclusion. So what's the delta G of this reaction? Products minus reactants. So the value of delta G will be negative. So anytime you see a reaction and it gives you a delta G for it, if it's negative, then that would indicate it's spontaneous. It's going to happen on its own. It's going to go from high free energy to low free energy. Here's an endergonic reaction. They require an input of energy. Here's the free energy of the reactants. Here's the free energy of the products. So we got to get up to here. That requires an input of energy. Is that spontaneous? No. It's not going to happen on its own without an input of energy. What's the delta G of this reaction? Products minus reactants. So the value of delta G will be positive in this particular case. So, here's a good summary of exergonic, endergonic that talks about entropy, delta G's, free energies. This is a slide you should study for your next exam. Okay? So exergonic reactions, their delta G is negative. They're spontaneous. They're downhill. They're going from unstable states to stable states. Or if you want to talk about entropy, they're going from low entropy to high entropy, organized to disorganized. What does this negative delta G value indicate? The maximum amount of work that the reaction can generate. This is the energy that's being generated by this reaction that can be used to do work. Why is the value negative? Because the free energy of the reactants is greater than the products. Endergonic is really just the opposite. Positive delta G's. They're not spontaneous. If you like, they're uphill. They go from stable states to unstable states. They go from states of high entropy or disorganization to states of low entropy. What's the delta G indicate? The maximum amount of work required to make that happen. The input that's needed. Why is it positive? Because the free energy of the reactants is less than the free energy of the products. So, we've got all these chemical metabolic reactions occurring in a cell. Would it be a problem if the cell let its metabolic reactions, particularly the ones that generate energy, the catabolic reactions, reach equilibrium? Yeah. Because at equilibrium, what's the delta G? Zero. That means there's zero energy available to do work. Well, cellular metabolism is organized in such a fashion that typically the metabolic reactions aren't allowed to reach equilibrium. How do we do that? That's because these metabolic reactions are involved in these multi-step pathways or conversions, where one thing is converted into another, that's converted into another, into another. The product of one reaction then becomes the substrate of a subsequent reaction. So each thing is converted stepwise into something else. The only way that the cell can pull this off and keep its metabolism from reaching equilibrium is if it's an open system. Remember what open system means? Open systems are able to do what? What can they exchange?
Oh, no, I was thinking something else.
Okay. Open systems are able to interact with their environment
I was waiting for it.
and exchange energy and matter. They can bring stuff in, they can take stuff out again. Okay, here's a little system. This is a closed hydroelectric system. We've got a reservoir of water here, and we've got a reservoir of water here, and connecting them is a little pipe with a turbine. Will this generate electricity?
For a little bit.
For a little bit, okay? Why? Because the free energy of this side is higher than the free energy of this side. So what's the water going to do? It's going to flow downhill, spin the turbine, and generate electricity. Well, what eventually happens? Reaches equilibrium. The two sides are equal. Turbine no longer spins. Work can't be generated anymore. So if this is analogous to a catabolic energy-releasing reaction, this could drive cellular work for a while. But eventually, it would stop, right? Here's a simple open hydroelectric system. Can this generate energy? Yep. Can it keep generating energy? As long as we keep putting inputs into this side and taking things out of this side. So cell metabolism is really kind of like an open, multi-step hydroelectric system. This is converted into this. This is converted into this. This is converted into this. But we keep needing inputs and we keep needing outputs to keep the system flowing. Overall, this is an energetically downhill flow. Therefore, it's spontaneous. Therefore, it's energy releasing. And we can continue to generate energy from this system. What does the water represent here? Food molecules the cell needs to take in. In the case of cellular respiration, something that's got a bunch of hydrogens on it, which are potential energy. What does this represent down here? The waste products that must be gotten rid of. The carbon dioxide, the water. Another really important point. These catabolic and anabolic reactions in the cell are oftentimes coupled together. They interplay with one another. Remember, catabolic are exergonic, anabolic are endergonic. So we can pair together an energy-producing reaction with an energy-requiring reaction. So some things that happen in the cell have to go uphill. Making fats. Glycerol and fatty acids are down here. Fat is up here. We want to make fats, it requires an input of energy. What do we couple that to? Something that releases energy, like the breakdown of glucose. The ATP cycle is a great example of coupling of endergonic and exergonic reactions. Oops. ATP hydrolysis to ADP and phosphate is exergonic. Higher free energy, lower free energy. That energy release can be used to drive endergonic cellular processes work. But pretty soon we run out of ATP.
Did you say that the exergonic is coupled with the anabolic?
Okay. Endergonic and exergonic, energy requiring endergonic, exergonic, energy releasing. Catabolic and anabolic really have to do with the same concept. Anabolic is energy requiring, catabolic is energy releasing. Okay? So the exergonic breakdown of ATP, which is catabolic, would be coupled to the endergonic synthesis of, say, a fat. Okay? Which is anabolic. Eventually, though, we need to generate more ATP. So sticking these back together again, that's endergonic. That's energy requiring. What do we couple that to? The exergonic metabolism of cellular respiration, for example. So it's really important to understand how these reactions cooperate together. So, here's an example. Here is a particular important amino acid. This is a different amino acid, and the cell needs both of those. The cell has the ability metabolically to convert glutamic acid into glutamine. But what does this delta G of this reaction tell you? It requires energy. It's not spontaneous. It's an uphill reaction. However, the hydrolysis of ATP is exergonic. Okay? It's energy releasing. So if I can couple those two reactions together, then the sum of that reaction is the sum of this and this. So overall, the reaction, if it's coupled to ATP hydrolysis, is exergonic. We need this much energy input to make it happen, but ATP hydrolysis produces this much energy. Okay? What if the delta G for this reaction was 8.8? Could ATP hydrolysis drive it? No, because 8.8 minus 7.3 would still be a positive value. Fortunately, ATP hydrolysis is a fairly large negative delta G value, which can then be coupled to chemical reactions in the cell that have a relatively small positive delta G value. We're going to see a lot of that in cellular metabolism. Here is something that we will be talking about, and you have a little bit of experience with in lab this week. Here's the disaccharide sucrose. Are we using sucrose? Okay. And one of the things you want to see in lab this week is, can yeast cells ferment this sugar? Fermentation is a type of cellular respiration. How does it differ from what I put up on the board up there? You still take sugar and convert it into carbon dioxide and water, and you're measuring the CO2 produced. What's the difference between fermentation and this type of respiration?
Oxygen isn't.
We don't use oxygen, okay? We're looking at a type of cellular respiration called fermentation. Okay? But for yeast cells to be able to ferment that sugar, the first thing they have to do is they have to be able to break this disaccharide down into these two monosaccharides. And then these can be used in the fermentation pathway to make the carbon dioxide and water and energy. Okay? But if you can't do this, you can't do fermentation. Well, the breakdown of sucrose into glucose and fructose is catalyzed by an enzyme. How do we know that's an enzyme?
It has ACE.
A good old ACE, okay, sucrase. Okay? So we're going to talk about the role that enzymes play now in metabolic reactions, and we're also going to talk about the role that they play in regulating metabolic reactions. So that's what we're going to do tomorrow, and then that's stuff that will be on the quiz on Monday. Okay?